That's not working. Okay, folks, let's get started. My microphone's not working right. So we get closer and closer to exam a week from Friday, and yes, I will do a review session before the exam, and I'll announce that sometime next week, most likely Wednesday evening, but I need to look at my own schedule as well for that. And yes, I do videotape that like I videotape the lectures. So we're a little bit behind, so I will keep moving things through. I want to finish up talking today about nerve transmission, and then we'll move our conversation to other important membrane considerations relating to the mitochondria. And I hear a lot of noise. Please be quiet. We'll move our conversation to the mitochondria. So last time I introduced the topic of nerve transmission, and I want to say just a little bit more about that today. So nerve cells are like other cells. They pump out sodium and they pump in potassium, and they do that because, of course, they have to maintain osmotic balance like any other cell does. One difference with nerve cells is that they have something we call ion-gated channels. And so I talked about ion channels before, and ion channels are largely selective pores through cells that are specific for allowing certain ions through. An ion-gated channel, I'm sorry, I shouldn't say ion-gated, a voltage-gated ion channel is a different kind of ion channel. It specifically is an ion channel that responds to changes in voltage. And so nerve cells do generate changes in voltage. They're known as action potentials. When you hear about an action potential, we're talking about a change in voltage across the nerve cell. And that change in voltage happens because of a stimulus. So that stimulus will vary depending upon what kind of nerve cell that we're talking about. Maybe it's a nerve cell responding to touch. Maybe it's one responding to smell or vision or something like that. It doesn't really matter. But in any event, the stimulus causes a first response. And the first response of a nerve cell is the opening of the sodium gates. That causes a whole chain of events to happen that results in ultimately the nerve cell telling the brain, hey, I got a signal. All right? So, as I said, the first thing that happens is the sodium gates open. That causes the sodium ions to flow rapidly into the cell because the sodium has been pumped out of the cell, so that the sodium concentration to start, that is at rest, is higher outside than inside. So sodium comes rushing in. That changes the voltage. And that voltage change causes the potassium gate at the same general location to open. So voltage change causes the potassium gate to open. Why does the potassium gate open? Well, the cell is trying to restore the ion balance, because remember, we had a whole bunch of positive charges rush in the form of sodium ions. If the cell opens the potassium gate at the same location, then potassium ions, which are higher concentration inside than outside, will flow out, thereby an attempt to neutralize the charge. So we see a pulse of voltage change. That's what's referred to as the action potential. And that also results at the very tip of that nerve cell in a change in the concentration of both sodium and potassium, because the cell's been letting in sodium and letting out potassium. Well, that disturbs the ion balance along the nerve cell. And when you disturb ion balances along a nerve cell, and you've got voltage-gated ion channels, these ion channels along the sides of the nerve cell start firing exactly the same way as the initial one did, which causes a wave of ion changes, a wave of charge changes, a wave of voltage changes along that nerve cell, and the nerve cell in that way is able to communicate, okay, ultimately with the brain or with another nerve cell or whatever. So that's how nerve transmission works. Now, one of the things I commonly mention is that there is a communication between nerve cells. And that's different from what I'm talking about here. What I'm talking about here is how one nerve cell gets a signal from one end to the other end. How does that nerve cell communicate with another nerve cell? It turns out that that's done through the action of what are called neurotransmitters. Neurotransmitters. Okay? A neurotransmitter is released by the signaling nerve cell. It binds to the next nerve cell, and it causes the same thing that happened in the first nerve cell to happen. Sodium gates open, sodium concentration changes, potassium gates open, we get a wave that goes through that next nerve cell. Okay? So because of this, depending on how many nerve cells we have to communicate a signal back to the brain or to the spinal cord, because of this, okay, information can be transmitted all the way from the point of the initial signal back to the brain center. Okay? Very important consideration. We can actually see this wave that happens in this one figure that I posted on the class site. Okay? I'm sorry, that's not the figure. That's just a text describing what I just described to you. You can see this signal moving along the nerve cell, obviously a lot slower than it actually is happening through the overall cell, through the nerve cell. This happens on the order of milliseconds. And because of the rapidity of this signal, we can, in fact, respond very quickly and carefully to the environment that our body finds itself in. Well, not surprisingly, nerve cells are pretty important for us to be able to respond and able to do the things that we do. So things that damage that ability to signal are very problematic. And one of those things two things that damage that ability to signal are two different neurotoxins that I mentioned here. One is called Tetrodotoxin, and no, you don't need to know the structure of that. But Tetrodotoxin is a neurotoxin, meaning it poisons/kills nerve cells. And it does that by binding to the sodium ion channels and blocking them. As a consequence, this guy kills nerve cells. This toxin comes from pufferfish. So pufferfish is a delicacy in Japan. It's eaten quite frequently, and there's one organ of the pufferfish that contains this tetrodotoxin. If it's not carefully removed, people get poisoned and can die from tetrodotoxin. Okay? So neurotoxins have very, very significant consequences if they're ingested. Another sodium channel blocking neurotoxin is called Saxitoxin. It's shown right here. It also binds to sodium channels, and it comes from certain algal blooms that can happen that are associated with various mussels. So you may hear periodically about the clamming season or other seasons on the Oregon coast that are closed, and they're closed because of a dinoflagellate bloom. That dinoflagellate bloom is producing Saxitoxin, and if people eat those mussels that contain that Saxitoxin, they can have some severe consequences, including even death. So that's what I want to say about nerve transmission. And I don't think there was anything, that just simply shows the ion gating right there. Okay, questions about that? Okay, let's turn our attention now to an important application of membrane transport, and that is electron transport. So I want to say a little bit about that to begin with. You learned something about electron transport in basic biology and also in cellular and molecular biology, and we'll probably talk about it in a little bit more detail than you've seen in some of those classes before. But I want to clarify something that students almost always confuse. Okay? Electron transport is the movement of electrons through various carriers in the inner mitochondrial membrane. Okay, I'll repeat that. So electron transport is the movement of electrons through various electron carriers in the inner mitochondrial membrane. The mitochondrion is absolutely, okay, The cell's power plant. You always hear that description. It's the cell's power plant. And it's the cell's power plant because of electron transport and oxidative phosphorylation. So we have electrons moving through these various molecules in the inner mitochondrial membrane, and as a result of that movement, okay, here's where students get confused, protons get pumped. They get moved across the membrane. Now don't confuse proton movement with electron movement. They're completely different things. There is no electron gradient. There is a proton gradient. The proton gradient arises as a result of the pumping of protons by the electron transport system. So the electron transport system is we can think of it as a circuit, okay? And that circuit causes something to happen. If I take, let's say, a small mechanical pump, and I put some batteries into it, the batteries provide, in this case, the circuit, and the circuit is letting electrons flow through the pump, and what's the pump doing? It's pumping, let's say it's pumping water out of underneath my house, okay? The electrons can cause movement of things to happen, and that's what's happening in electron transport. It's something's being moved. In this case, the protons are being moved. Okay, well, before I talk about that, I want to just introduce some general topics. I want to go through these very quickly. I'm sure you've seen some of them before. One is I've mentioned that, of course, oxidation is the primary energy source for cells. And we saw examples of oxidation happening through the citric acid cycle. We'll see numerous other examples of oxidation during the term. When we think about oxidation, we think about something that starts at the most reduced state, that is basically carbons and hydrogens, and getting to the most oxidized state, which are basically carbons and oxygens. And if we look at the various energies associated with each of these molecules, we discover that there's more potential energy, as shown by the size of that negative sign here, the negative delta G 0 prime kilojoules per mole, of the more reduced molecules compared to the more oxidized. So this can generate more, this guy here can generate more energy than this guy, which can generate more energy than this guy, which can generate more energy than this guy, which can generate more energy than this guy. These are various stages of oxidation, and cells control oxidation enzymatically. We talk about redox reactions. It's very important. I don't know why chemistry isn't very good at teaching students this, but I will repeat it because I find students frequently overlook this. For every oxidation, there's a reduction. Now, if I say that to you, every person here can repeat that to me just like that. But learning that and understanding what that means is a different thing. I want you to understand that for every oxidation, there's a reduction. What does that mean? All right? Well, what it means is oxidation is the loss of electrons. That should have come through in chemistry. Not your fault, it's chemistry's fault. They don't teach you that well, okay? Oxidation is the loss of electrons. If you lose electrons, they don't disappear. Matter is not created or destroyed in chemical reactions. Okay? So what happens to the electrons? Well, they go to something else. They don't evaporate. They don't disappear. So if they go to something else, that means something else must gain electrons. And that's why, for every oxidation, that is a loss of electrons, there's a reduction because something else is gaining those electrons. Now, biological systems are very different. Okay? They're very different from chemical oxidations that are happening out here in the environment. Okay? If I take a match and I light it, what happens? I see a flame. I generate a tremendous amount of heat, right? And if I do that with a cell, what happens to the cell? It's fried. Cells have to control oxidation. And they control it very carefully using electron carriers. So in this example that I show you on the screen, I have a molecule on the left that is being oxidized. It's going from this guy right here, which is an aldehyde, to an ester. An ester comes from making an acid, all right? So we have aldehyde to acid, essentially. That's an oxidation. Well, if we have an oxidation, that means loss of electrons. This guy had to lose electrons, and if I'm doing a chemical reaction out in the real world, those electrons go to the first molecule they bump into. Cells don't like that. Cells are control freaks. They have to be control freaks because they can't have that flame going off inside of them. Cells use enzymes to pass electrons to electron carriers. The electron carriers you've already seen. You've seen NAD gains electrons and a proton to become NADH. Or FAD gains electrons and 2 protons to become FADH2. Okay? Those things, all right, are reductions. So you can see very clearly in this equation that the oxidation is coupled to a reduction. And yes, this reaction is reversible. If I move from this guy over to this guy, that would be a reduction. Reversing an oxidation is a reduction. That means I have to have a source of electrons, and guess what the source of electrons would be? This guy right here. NADH going back to NAD would be an oxidation. Okay? Now, because of this ability of cells to control electrons in this way, energy changes that happen as a result of oxidation are manageable, and the cell doesn't catch on fire. And the cell can use that electron energy that's contained in those electron carriers to do useful things. Now, I like to think of this process, okay, and I'll describe it in a couple ways, as charging a battery. If you charge a battery, you can use the energy of that battery to do something. And we're going to charge the battery in a couple ways. You could think of one way of charging it as putting those electrons and proton onto NAD to make NADH. You'll see there's other things that can use. You can use one battery to charge another battery, right? How many of you have the little boosters for your phone that you can plug in and it will charge your battery? That's just a little battery in here that's charging your phone battery. The battery of NADH can be used to charge your mitochondrial battery. And that's what's going to happen in electron transport. Okay. NADH, NAD. Memorize those structures for the exam. No, we're not going to memorize those structures for the exam. Okay. All right. It got very quiet. I didn't hear nobody said, no! Right? You should not let your instructors do that to you guys. You know, if you don't say anything, I might think, oh, they're okay with this, right? Let's memorize these structures for the exam. Here's 300 of them for the exam. Let's memorize all those. All right. I just show you this to show you what this reduction looks like this oxidation looks like going from left, the reduced form, to right, the oxidized form. Okay? Loss of electrons is an oxidation. Okay. The ultimate source of all the battery charging, the ultimate source of all the energy on the face of the earth, essentially, is photosynthesis. Okay? The ability of plants to capture energy from the light of the sun is remarkable. And we don't talk about photosynthesis in this class, unfortunately, because we focus more on animals. All right? But photosynthesis is very, very similar to electron transport. In some ways, it's like electron transport in reverse. And you say, well, how does it go in reverse? When we think about electron transport, what's driving electron transport? Oxidation. Oxidation is driving electron transport because it's providing the charged battery in the form of reduced electron carriers. If we reverse that process, okay, we actually make reduced electron carriers. What's the energy source then? The energy source is the sunlight. So the sunlight provides that backwards. And we won't talk about that in class, but I want to make you aware of the fact that photosynthesis is a sort of reversal of the electron transport process. The oxidation and reduction happens as a result of catabolism and anabolism. Okay? We can take small molecules and build them into bigger molecules if we have a source of energy, and that source of energy comes from the hydrolysis of ATP to make ADP. Where do we get the ATP from? We get the ATP from oxidation. So if we want that process to move upwards so that we can make larger molecules from smaller molecules, proteins from amino acids, nucleic acids from nucleotides, fats from fatty acids and glycerol, carbohydrates from sugars. If we want to do that process on the top, we've got to have these processes on the bottom occurring. Otherwise, we're dead. Okay. Now, these processes happen across the membrane of the mitochondrion. This is something I've sort of shown you before, because of differences in concentration of chemicals and differences in concentrations of ions. Now, there is some mathematics associated with that, and here's the good news for you. I'm not going to make you responsible for the mathematics of this, okay? But it will suffice to say that there is an electrochemical potential that you can actually measure associated with these reactions that are not the reactions, I'm sorry, associated with these concentration differences across the membrane that we can actually measure. And those electrochemical differences give rise to something called the Nernst Equation. Okay? No, we're not going to do that calculation. All right? But the Nernst Equation tells us the relationship between the electrochemical concentrations and the potential energy in terms of voltage that they give rise to. Okay? I can plug that information into an equation and derive a delta G that corresponds to the electrochemical differences across the membrane. We're not going to do that, but suffice it to say that if we have an electrochemical difference across the membrane, there is an energy associated with it, and like any energy, okay, we can relate that to the free energy change that's important for the direction of reactions to move. Okay? If you know that much, you know all you need to know about the electrochemical differences. Well, let's talk about the mitochondrion. The mitochondrion is an amazing structure. Okay? The mitochondrion, of course, is the energy power plant of the cell. The mitochondrion is an organelle of eukaryotic cells. We don't find mitochondria in prokaryotic cells. We don't find mitochondria in prokaryotic cells. Prokaryotic cells have to use some tricks to do the things that eukaryotic cells do in their mitochondrion. Okay? And yes, prokaryotic cells do use gradients to make ATP like we do. But because they don't have things sequestered nicely in an organelle like we do, prokaryotic cells have to use those tricks. We're not going to go into those tricks. But exactly the same thing that happens in mitochondria happens in prokaryotic cells for the generation of energy. Okay? There's pretty good evidence that the mitochondria that we see in eukaryotic cells originally was an independent living cell. And it got gobbled up by a bigger cell. And over time, they formed a symbiotic relationship. Okay? That relationship had the cell, the bigger cell, providing nutrients to the mitochondrial cell, the mitochondrial cell using those nutrients and generating ATP. So that symbiosis eventually, okay, was so strong that mitochondrial cell lost most of its function. Okay? The important function that it had about providing ATP was maintained. And that's critical for our cells. We have evidence that the mitochondrial cell actually was originally a cell because the mitochondrion contains its own genome. It's one of only 2 cellular organelles that actually contain its own DNA. The DNA that's contained in that mitochondrial cell is fairly simple. In human beings, it's about 16,000 base pairs long. Okay? It codes for some proteins that the mitochondrion uses. And when we look at the sequence of that DNA, and we look at the way that DNA is coded to make proteins, it looks a lot more like bacterial DNA than it does eukaryotic DNA. It's not proof, But it's consistent with the idea that this was originally a bacterial-like cell that got engulfed and taken in. Okay, well, that's more than I need to say. The other organelle that has its own genome is the chloroplast. Okay, it's the chloroplast. And the chloroplast is a little bit more complex in the DNA that it contains than the mitochondrion is. But just like I told you that the photosynthesis is sort of the reverse of the electron transport system, not surprisingly, the chloroplast also has its own genome, and it probably also was a primitive cell that got gobbled up by a bigger cell. Okay, well, let's look to see what's happening here. This is a kind of a complicated slide. I'm going to step you through it. All right, here's the mitochondrion. The mitochondrion has two membranes. It has what we call an outer mitochondrial membrane that doesn't do a whole lot. It's relatively porous, meaning it has holes in it that can allow molecules of up to about 50,000 in molecular weight pass through it reasonably readily. I like to think of the outer mitochondrial membrane as kind of a wet blanket. You put a wet blanket over something and things can pass through it, but it's going to be kind of cumbersome for them to do so. That's kind of what the outer mitochondrial membrane is like. The inner mitochondrial membrane, okay, is the suit that a frogman wears underwater. It's tight. Molecules, okay, for the most part, do not readily move through it. It's what we call intact or integral, okay? It maintains its integrity. And its integrity turns out to be absolutely critical to the function of mitochondria, as we're going to see over and over. Okay? What does integrity mean? It means no leaks, no holes, no runs, no drips, no errors. The integrity of the inner mitochondrial membrane is essential for its function. Here's what's happening in a cell, in a eukaryotic cell. Out in the cytoplasm, we have glycolysis occurring. You learned that last term. And here's glycolysis going from glucose all the way down to pyruvate. That all happens in the cytoplasm of the cell. In order for the reactions of the for the pyruvate to get to the citric acid cycle, it has to be transported into the mitochondrion. And there are transport systems that do that, and the reason that's important is because the citric acid cycle is occurring inside of the mitochondrion. Excuse me. The mitochondrion has some structural features that we describe. I already mentioned the outer mitochondrial membrane, the inner mitochondrial membrane. The inner mitochondrial membrane is very folded upon itself. This is not very, in fact, the reality is these folds that you see here come way in. It's very, very convoluted with folds, and the more folds you have, the more surface area you have for the inner mitochondrial membrane. The folds are important for that because surface area turns out to be very important because it's in the inner mitochondrial membrane where the electron transport system is located. Now, you probably don't remember when I showed you the composition of various membranes the other day in terms of proteins. But I will tell you that the membrane that has the greatest concentration of proteins is the inner mitochondrial membrane. This underlines the importance of what the inner mitochondrial membrane is doing and the importance of these proteins in getting molecules and other things in and out of the mitochondrion. Okay? So these infoldings that we see that increase the surface area of the mitochondrion are very important for mitochondrial function. The inside, what I like to think of as the soup of the mitochondrion, this is the liquid portion on the inside. It would be the mitochondrial equivalent of the cytoplasm, although it's not the cytoplasm. We'll call it the mitochondrial soup. This region of the mitochondrion is called the matrix. The matrix is the liquid portion of the mitochondrion, the far inner part, and it's in the matrix that the enzymes of the citric acid cycle are dissolved, with one exception. And that one exception I mentioned last time is succinate dehydrogenase, which is embedded in the inner mitochondrial membrane. We'll see more about that in just a bit. All right, so what's happening? Glycolysis produces pyruvate. Pyruvate gets moved into the intermitochondrial membrane. Gets moved into the mitochondrial matrix. In the matrix, the citric acid cycle occurs. And in the citric acid cycle, we have production of reduced electron carriers as a result of oxidations. The oxidations of the citric acid cycle drive the production of NADH and also of FADH2. Okay? Well, the cell and the mitochondrion, as part of the cell, have limited amounts of NAD and NADH. You know that NAD is essential for oxidation. What happens if the cell runs out of NAD? Well, we talked about that before. Oxidation is going to stop because there's no place to put electrons. Okay? So the cell has to have a very quick and easy way of regenerating NAD from NADH, and that's what the electron transport system is actually for. In the electron transport system, NADH dumps its electrons off to the electron transport system. And when it does that, NADH becomes NAD. Doesn't get any simpler. Well, it's important for that to happen. Does the electron transport system have infinite capacity? Yes and no. Okay? The electron transport system has to do something with those electrons. It passes them from what we call one carrier to another, and I'll show you that, sequentially, like that electronic circuit that I mentioned. Okay? But those electrons have to go somewhere, just like those electrons from your battery that are driving that pump have to go somewhere. Okay? Anode to cathode, right? Or cathode to anode, I never can remember that, right? They're going one direction or the other. They have to end up somewhere. Well, those electrons in the electron transport system end up somewhere, all right. They end up in an electron acceptor. And the electron acceptor that mitochondria use is oxygen. So when I say cells have an infinite carrying capacity for electrons, they do if they have an infinite supply of oxygen. But if they don't have an infinite supply of oxygen, what's going to happen? The system's going to get plugged up, right? And if the system gets plugged up, what happens to the conversion of NADH to NAD? Anybody? It's going to stop. And when that stops, what happens to citric acid cycle? It stops. When citric acid cycle stops, what happens to glycolysis? It may stop. Okay? We got problems. Not really problems, it turns out. These are actually a normal part of what our cells are doing all the time. As I mentioned before, you're sitting out here, you're not actively exercising right now, except for perhaps the cells in your brain. Your muscle cells aren't doing much. They're probably not doing much oxidation. They probably have an abundant supply of oxygen, right? So what's happening here that this isn't going crazy if you have an abundant supply of oxygen? Well, we're going to see that there's a variable that I haven't talked about so far that determines whether or not electron transport gets to run. I'm going to save that for you for just a little bit, okay? But it's part of respiratory control. For our purposes right now, we need to understand the movement of those electrons. So let's focus on the movement of those electrons through the electron transport chain, okay? This schematically shows what's happening inside a mitochondria. We can see, for example, that electrons from NADH up here are being donated to something that has this Roman numeral 1. This thing that has a Roman numeral 1 is a protein complex that's found in the inner mitochondrial membrane that has a name that's about this long that we're not going to know. I couldn't repeat it to you if I had to. We're going to call it complex 1. Nice and simple. That's what the 1 call is referred to. Electrons from NADH get donated to complex 1, and that generates NAD. Complex 1 donates electrons to something that's also embedded in the mitochondrial inner membrane, and it's a little tiny molecule that's called coenzyme Q. Coenzyme Q. Coenzyme Q turns out to be an interesting and important molecule. Okay? Electrons donated to complex 1 are donated in pairs. They always come as 2. Complex 3, which is the next site after coenzyme Q, can only accept electrons one at a time. Well, something has to manage the electrons and coming in pairs and going out as one, and the thing that does the managing is coenzyme Q. You can also call it CoQ, C-O capital Q. Okay? So CoQ donates one electron at a time to complex 3. You might wonder where complex 2 is, and I'll come back to that. Complex 3 donates one electron at a time to the next carrier, and it's a protein called cytochrome C. You may have heard of cytochrome C. Cytochrome C is a protein that's found all the way from bacteria to human beings, which says something about its importance when you see something so widespread. Cytochrome C accepts electrons from complex 3 and passes them off. It accepts them one at a time, and it passes them off one at a time to something called complex 4. This is very simple. NADH to complex 1 to coenzyme Q to complex 3 to cytochrome C to complex 4. There's only one other place electrons go. And in complex 4, electrons are donated to oxygen, which gains some protons and makes water. It's because of this last step that you start breathing heavily when you exercise. It's because of this last step that if you run out of oxygen, you're going to have troubles. Because if you're not out of oxygen, you've just gummed the system up. Okay? And if you gum the system up, then you can't regenerate NAD from NADH. And if you can't regenerate NAD from NADH, then your NADH concentration is going to increase, your citric acid cycle is going to stop, your ability to produce ATP is going to stop, and you do that long enough, and you're going to die. We don't want to gum the system up. But gumming the system up turns out to be a part of what cells normally do, and that's the respiratory control I've talked about and I'll talk more about later. Okay, one more thing about all of this that's really cool that I haven't said, and that is that the movement of electrons through complexes 1, 3, and 4, each time electrons move through one of those complexes, protons get moved. They get pumped. I call it pumping. They get moved from the mitochondrial matrix out into the intermembrane space. That's the space between the inner mitochondrial membrane and the outer mitochondrial membrane, the wet blanket. Can protons get out through the wet blanket? Yeah, they probably can, but for the time that the wet blanket is sitting there, they're probably holding it fairly close to the inner mitochondrial membrane. What does that mean? Well, as a result of this pumping, the concentration of protons in the matrix is decreasing, and the concentration of protons in the intramembrane space is increasing. And we have just made an electrochemical gradient. We've just made an electrochemical gradient. And we know that associated with electrochemical gradients, we have electronic potential, and that electronic potential gives rise to energy that cells can use. Now, I haven't said yet how will the cells use that. I've simply said that we've created this gradient. Okay? Let me say another thing about electron transport. I've shown you one system. There's another system. Another system is actually very much like the first system, except for it uses something called complex 2 instead of complex 1. Well, how does complex 2 get into the picture? Complex 2 accepts electrons from FADH2. Remember, we also generated FADH2s in the citric acid cycle. And where did we generate electrons in the citric acid cycle? Through succinate dehydrogenase. And succinate dehydrogenase was the only enzyme of the citric acid cycle that was found in the inner mitochondrial membrane. Guess what? Complex 2 and succinate dehydrogenase are the same thing. Now, this turns out to be very convenient, okay? Because what happens is complex 2 simply donates its electrons to coenzyme Q, which donates them to complex 3, which donates them to cytochrome C, which donates them to complex 4, which donates them to oxygen, making water, just like the other system. The only difference is electrons are going through complex 2 instead of going through complex 1. Now, if you think about this, there is a difference. And the difference isn't just the fact that one's using complex 2 and one is using complex 1, but let's look at what happened in the first case. Electrons coming from NADH went through complex 1, which involved proton pumping. Okay? I'm sorry, right here, complex 1. It involved proton pumping. Complex 2 does not involve any proton pumping. That means that if we compare the number of protons pumped per FADH2 and compare them to protons pumped per NADH, we would expect there would be more protons pumped as a result of electrons coming from NADH, and you would be exactly right. Go to the head of the class. Okay? People make estimates in terms of how much energy is generated as a result of the pumping of the protons. And people get really wound up about counting how many protons and is it exactly this number, etc., and we don't really care. If you use the estimate that about enough energy to make 3 ATPs is generated by electrons from NADH, you're in the ballpark. About 3. It's not exact because protons can do a variety of things. They don't have to all go to make ATP. But about 3 ATPs resulting from electrons from NADH. And no, I haven't said how the ATPs are made yet. I will. By contrast, ATPs from the movement of electrons from FADH2 corresponds to about 2 ATPs. Okay? If we use those estimates, and people argue about them, whether it's 2.5 or it's 3 or it's 2 or whatever, it doesn't really matter for our purposes. If we use the estimates of 3 and 2, that's where we get the numbers that say that the complete oxidation of glucose to carbon dioxide results in production of about 38 ATPs when oxygen is present. Oxygen has to be present for this whole process to happen. If oxygen is not present, then the cell cannot do this process, and it relies on fermentation to generate those ATPs. And those ATPs generated in fermentation, there's only 2 per glucose. So this generates 19 times as much energy when oxygen is present as when oxygen is absent. Okay. Clear as mud, right? Quiet group. Questions? Yeah.
So I don't know if it's just because of the diagram, but if electrons go through complex 2 for the second system,
Yep.
why is there still a complex 1?
Well, it's there. The complex 1's everywhere. The complex 1 is there. It's just, in this case, I'm showing it's bypassing complex 1. If you were to look in this mitochondrial membrane, you would see thousands of complex 1s. Okay? Now, these carriers that carry the electrons, okay, are molecules, okay, that contain iron or copper. All right? You saw what heme did in the carrying of oxygen in your blood cells last term. Okay? Hemes make up some of these molecules. The difference between an iron plus 2 and an iron plus 3, that is ferrous versus ferric iron, is 1 electron. And this is why heme and iron, okay, are important electron carriers. Because an iron can accept an electron, and then it can pass it off. And it can accept it, and then it can pass it off. The last heme that's in complex 4 is the only one that contains copper. It's the only one that contains copper. All the other hemes contain iron. And what can copper exist as? Copper can exist as cupric, cuprous, if you remember your freshman chemistry. And that corresponds to a +1 for copper, cuprous, or a +2 for copper, cupric. Again, a difference of 1 electron. So we can see the movement of electrons through these systems by the changing charges of the irons and the coppers. Okay? Make sense? All right. Well, the last thing I want to say then about this figure actually relates to this thing that's in the yellow box up here. I'm going to say more about it next time, but I'm going to start to talk about it here. It's a related process to electron transport, but please understand it's completely physically separate. It's physically a distinct system from the electron transport system. This is what we call oxidative phosphorylation. And oxidative phosphorylation, okay, is using the electrochemical gradient created by electron transport to make ATP. We charge the battery by pumping protons out, and now we're going to use that battery to make ATP. The making of ATP by that battery corresponds to oxidative phosphorylation. Okay? How does it occur? Well, schematically you can see on here it occurs very simply, and it turns out to be it's kind of a cool process. All right? The electron transport system creates a gradient of protons. Those protons want to come in. If I take water with my sump pump and I pump it up into my attic, I have a high concentration of water. It wouldn't be a good move, but I have a high concentration of water in my attic. What does that water in my attic want to do? It wants to come back down, right? So if I let that water in my attic come back down and I let that water turn a turbine, I could use the energy of that movement of the water to do something. Charge another battery, right? Well, the movement of this water, in this case the protons, back into the matrix is turning a little turbine, and it really is a little turbine. I'll show you that next time. The movement of that turbine causes ATP to be made from ADP. And that's the way that 99% of your ATP in your cell is made. By the turning of a turbine. Amazing. Okay, that's a lot of information. I think we should have a little song about that. And this one requires your help, seriously your help. It's a song I think you're going to know. It's called If You're Molecular and Know It, Clap Your Hands. I'm going to sing the parts in black, you're going to sing the parts in red. Are you ready? If you want to have a lot of energy, you had better make a lot of ATP. I will only tell you once. You need proton gradients and a bunch of starting stuff like ADP. If you hanker for a sweet that you can taste, and your Atkins diet book has been misplaced, you should know adrenaline is an aid to getting thin. Putting phosphates onto enzymes trims your waist. If you're feeling kind of achy in your ways, and that hangover has hung around for days, you should know, you silly dear, pain does not come from your beer. Prostaglandin is made by PGH synthase. H-synthase. There are acids in the bile that make up gall, which emulsify acetylglycerol. If your health has gone to hell, you should blame the LDLs because they carry all of that cholesterol. Some phosphates and a sugar on a base make up C's and G's and U's or T's and A's. You can make a DNA or a strand of RNA if you add a template and polymerase. If you want to ace this test with utmost ease, you don't really have to get down on your knees. And you need not say a prayer, so please don't pull out your hair. Just go download QuickTime and the MP3s. All right, see you guys on Friday.